differentiation process. So till here, I'm sure that all of you have understood whatever we had discussed. We are talking about potency. These are the totipotent cells. Again, when we are coming to the next stage, that is blastocyst stage, we'll see that some cells which are inside. Okay, these cells are called as embryoblast cells. Now, when we take out these cells, they become pluripotent cells. Pluripotent cells are the ones which are capable of giving rise to various systems. Okay, they are capable to give rise to various different type of systems. So that's why these are called as pluripotent stem cells. Now you keep one cell here into a culture media and give rise to cardiac muscle developing factor. Okay, so it will give rise to heart and blood vessels. If you keep nervous system nerve growth factor, it develops into nervous system structures. If you keep into immune system growth factors, it will develop into immune system structures. So that's why these cells are called as pluripotent cells. Now these cells, okay, once they are entering into a particular region, they become multipotent cells. Okay, multipotent means they can give to the muscle cells which are forming circulatory system. They can give rise to epithelial tissue which is giving rise to circulatory system. They can give rise to connective tissue which is giving rise to circulatory system, like that. Okay, so these cells are called as multipotent stem cells. And finally are the unipotent cells. Unipotent cells means they are more differentiated cells. Now once they are destined to form a cardiac muscle cell, now they're not going back. Okay, you can't cultivate in any type of growth factor, they'll not give you anything. They will give you only cardiac muscle cells. So this type of single potency, okay, they have only single potency. That type of cells are called as unipotent stem cells.
चायमध्ये साखरच नाहीये.
Please mute yourself. So you will see that these are the various capabilities, potency of a cell, embryonic cell, and how they are differentiating. Okay, so that's about systems. Now we have to learn about a term called as apoptosis. Already we discussed that. Okay, what is apoptosis?
Ma'am,
Yes, Harsh.
ma'am, can we then? It means we can produce many children from single pregnancy.
Yes, you
That
can.
if we
You
take
can.
many cells from morula stage.
Yes, that's why that is banned. You know cloning process? Have you heard of cloning?
Yes, yes,
What is cloning? What
yes,
they
ma'am.
do? We take all types of genes, okay? They take oocyte and sperm, fuse them, okay? And they genetically manipulate the genes of a zygote. So what they do, one single cell they take out, they manipulate the genes which are present in this single cell. What they do, they want, acha, for example, I want a blue-eyed baby. Okay, I want a boy. I want a boy child with who will grow to 5 feet 9 inches. Okay, I want a boy child who is very slim, handsome, and with a very high intelligence IQ levels. I can manipulate all that because all the genes which are responsible for color, height, weight, everything, we know that. Okay, we know the genetic map, blueprint of the gene, we know it, of the DNA. So what I do, I manipulate whatever is what I require, and I will introduce that genes only what is required, or else I can make him like an Superman or Iron Man who is very strong. Okay, I can do whatever I want because I can manipulate all the genes, and I'll develop this single cell into an entire organism. And I just need to, what I have to do, I just need to introduce this back into the uterus. It will start growing. So this process is cloning process. That is what is, that's why the world scientists have banned this, because yeah, if we are trying to develop only we cannot get only good things out of it, right? We can create Osama bin
Yes, ma'am.
Laden's also, right? So that's why they have stopped. Okay, cloning process is officially banned at many
Ma'am,
places. Yes.
isn't it better to change certain aspect of a human body? Like if we want a tall child, then
Mm-hmm.
we could manipulate it, no? So
Yes,
why
we
it
can
is wrong?
manipulate. Yeah, we can manipulate it. Now, you are happy with the child who is tall, but I am not happy with just a tall child. I want my child to be super intelligent. Okay, IQ level which is
Yeah,
greatest.
so what is wrong in that, ma'am?
Now maybe there might be my brother who wants a child who is very crooked, very dangerous. Okay, who has always intention to destroy everything, right? Or else maybe someone else might say that I want another Osama bin Laden. We can do that, isn't it? We are not, see, human expectancies will not stop at one level. We want more always. So you and me might be happy with smaller things like a fair child, or we might say that yes, the child should be a boy, or we might say that yeah, the child is a, I want a curly haired child. We might be happy with only that, but imagine if we can do this genetic manipulation. To what extent we can manipulate? We can do anything, right? So that means there is, yeah, yeah, Juhi.
Ma'am, in pluripotent,
Hmm.
it give rise to various system, right? But
Yes.
not another embryo.
No, no, not another embryo. The total potency will
Okay.
grow. Okay. So that's why genetic manipulations are very, very difficult, very, what to say, not difficult, they are dangerous. Okay. So that's why, see, that's why the cloning process is only permitted to only rarest of the rare species of animals right now. The animal species, okay, that's why if you see anthropological studies, they go and find out the species, rare species genetic, the DNA which is available, they try to recreate those organisms. Okay, so that's why very few only it is permitted to animals, that too only very rare species all over the world. So everyone has banned this process because of it can lead to greater destruction rather than good things. That's why imagine if all of you are intelligent. Okay, let's say 100 students in your class, all of you are very intelligent. Now all of you, once you clear out, how many jobs are required? Will you have that? And you may not have that many opportunities. Now one of you might think that okay, I'm happy with whatever I'm doing. Another might think I want more. For getting more, maybe we'll go for destruction. Then what we'll do? We'll fight with each other, isn't it? To possess things, because we're all intelligent. So nature has a balance, that some are predators, some are weaker animals. Why? Because there should be a cycle maintained. So same like that in humans also, some are capable in doing certain things. They are technically good. Some are like, now you take for example a medical school. Some teachers are excellent in teaching. Some teachers are very good at surgical procedures, but they may not be good teachers. So like that, we have various people, different, that's the beauty of nature, that we are all different in different things. We are good at different things. So that we are all having our own area of specialization where we are good at. Imagine everyone are good at everything. Now where does the opportunity come from? isn't it? So that's why genetic manipulations are bad. So that's why the cloning is banned. Okay. I hope all of you are clear with potencies.
Yes.
Yes, ma'am.
Now let us talk about apoptosis. Apoptosis is what? Apoptosis is called as programmed cell death. For example, what happens is, now see in this picture, you can see that this is how a first initially, this is an electron microscopic picture, which is original, and you see that the limbs are growing like this. Okay, initially you will see that it's all webbed. Okay, webbed hand. Now slowly, webbed means like a web. You can see that the two fingers are connected with each other by a web. Okay, you see like a duck. If you see duck limbs and all, okay, they're all connected, webbed. Now what happens is, later on, as they grow, these cells which are present in between, this is not required. We don't need a webbed hand, isn't it? For us, what is required? We are not we don't want more, what to say, balance while walking, because we are not using our upper limbs and lower limbs for especially upper limbs for walking. We are two-legged animals. Okay, right? So what happens is, our upper limbs are first to take out apoptosis, to initiate apoptosis in the upper limbs. So what happens is, the cells which are present in between, they die, giving rise to nice spaces between the fingers. So when these spaces are coming, what we'll get? We'll get nice separate fingers. So this process of programmed cell death, after a certain time, the cells die. Okay, so this is called as apoptosis. Even our RBCs, what is the time limit for RBCs? 120 days. After 120
120
days,
days.
yes. So after 120 days, how do they know that they have to die? They are programmed like that. So that's why they once their time is over, they die. So this process is called as apoptosis. Even any diseased cell in our body, it dies. Why it dies? Because it is programmed like that. If it is not fine, it has to die. Okay, so cells die in response to a variety of stimuli. Now there should be a stimulus. When that apoptosis has to start, there should be a proper stimulus to it. Okay, and this apoptosis is also a very controlled, regulated procedure, not just anytime it happens. It is a very controlled mechanism. See the beauty of human organism. I still tell many people, no engineer can make a machine like human body. Such a beauty it has. Such well organized, synchronized process. Okay, so that's how our limbs are formed. That's how very beautifully everything is the mechanism, everything is organized. It is present in a proper time. Okay, and proper way. That's all happening in a regular fashion. Okay, so this process is called as apoptosis. Now we already discussed about cell potency, what is totipotent cell, pluripotent cell. Okay, already we discussed that. To add to that, okay, it will give rise to any cell.
Ma'am,
Yes.
ma'am, your voice is lagging.
Okay, um, is it still lagging?
No, ma'am.
Okay. Okay, pluripotent cell, already we discussed that. Okay, it can differentiate into any fetal or adult cell type, or any of the three germ layers: endoderm, mesoderm, ectoderm. We'll discuss this bit later. What are these three germ layers? Right now just understand what are the types. We discuss this. Now, multipotent progenitor cells, examples given here are hematopoietic stem cells, the cells which are giving rise to blood cells. See here, hematopoietic stem cells, HSZ, Z0. Okay, so from here they might give rise to different types of differentiation processes, you can see. Okay, it will give rise to L blast, mo blast, pro M, myeloblast. Okay, so like that, pro megakaryocyte, so like that. So various types it can give rise to. One cell can give rise to various type of cells, and from there, now these are very differentiated ones. Okay, these are stem cells, these are differentiated cells. Once the differentiation is done, it will give rise to only one specific type of cell. Okay, so these cells are unipotent cells. Now these cells are multipotent cells. Okay, these are unipotent cells, okay, or precursor cells, hepatocytes. Only one cell type we are getting. Okay, these are the hepatocytes which are present in the liver. Hepatic refers to liver. Okay, liver cells are unipotent cells. Whenever there is an injury, the cells start regenerating. So liver has got regeneration capacity, right? So that's why we even when we are going for transplants, liver transplants and all, small portion of liver is sufficient for transplantation, because some of the cells regrow. Okay, they have a regeneration capacity. So this type of capacity is called as unipotent cell or unipotency of a cell. Now let's see some terminology related to it. Ovum. Ovum means what? It is the meaning egg. Okay, it's a Latin word meaning egg. Ovum means. Sperm is derived from Greek word sperma, means seed. It's a it is derived from the word seed. Okay, sperma. What is a zygote? Zygote is a cell that results from union of oocyte and the sperm during fertilization. So this is the stage where you get oocyte and the sperm. Once it enters inside, you can see two nuclei here. One is male and one is female pronucleus. So this stage is called as zygote. Now, what is an embryo? Now, embryo is 3 to 8 week of the development. Okay, so at least 1 to end of second week, we call it as zygote. From third week after implantation onwards, we will call it as an embryo. Okay, till when? Till 8th week. Now, from that, we also use conceptus. What is conceptus? Conception. Conception means pregnancy. Conceptus means the embryonic tissue. Now, what is the embryonic tissue? All the developing embryo. This is a developing embryo, and all the structures related to the developing embryo. Okay, this is when usually after delivery, after parturition, along with the developing embryo, we need the fetus. Along with the fetus, we need all the tissues of the related to the fetus should come out during pregnancy. Especially we will see all this sac, chorionic sac, amniotic sac, so the fetal membranes, placenta. So all these are the fetal structures which are present inside the uterus during pregnancy, during delivery or the labor. It has to come out. Okay, so this all tissues are called as conceptus. Conceptus means what? It contains embryo as well as all the structures related to the development of the embryo. Okay, primordium. What is the word primordium? Or it is also called as analgae. Analgae, Analg, analgae. Okay, analgae or rudiment. So what does that mean? This is the first discernible indication of organ or a structure. So see here, while development, if you take an embryon, electron microscopic picture, this is how a developing embryo is present. See, you can see the developing face, neck, heart is here. You can see the liver bulge. See the limb buds, you can see the limb buds. Okay, so these are the upper limb buds. We'll see lower limb buds here. Okay, so like this, these are called as primordia. Okay, I'll say that this is the primordium of upper limb, this is the primordium of lower limb, this is the primordium of heart. Okay, primordium of the brain. So when we are telling this primordium means what? These are the first indication of a organ or a structure. Now, what is a fetus? Fetus is an unborn offspring. Okay, it is still not yet born. It is usually we start this fetus word from 9th week till the birth. Okay, embryonic period. So till here, this is called as fetal period. Okay, this is the when we call it as embryo from third to eighth week, that is called as embryonic period. This is called as fetal period. Okay, now there are certain planes where we take sections, same like adult. We also take various sections inside the and in the fetus also, in the fetal tissues also. Remember that without understanding the section, you will not get a clarity on how we are looking at this developing organism. It's very very complicated. Okay, so especially embryo, whenever you are looking at a picture, please understand which type of section you are looking at. Then only you will get a clarity related to it. Okay, so now see here, what are the various words we need to understand in an adult body. This is superior, inferior, anterior and posterior, which we have already studied that. Now in a fetus, we use these words: cranial, cranial end of the fetus; caudal, caudal means tail end. Okay, head end, cranial, caudal; ventral, ventral is the anterior side of the fetus; dorsal, okay, dorsal side, posterior side of the fetus. So these are the words: cranial, caudal, ventral, and dorsal. So these are the various words. Then see the sections. What are the various sections? We see that we can take midline section, okay, or it is also called as median section. Sagittal section, already we discussed that sagittal section divides the body into two halves, right and left half. So the fetal sections are quite similar, but when you are looking at the structures, okay, the section which you are taking in the fetus and the section which you are taking in the adult are quite different. Okay, you will not understand the same structures which you will see in the adult will not be there in the fetal tissue. Okay, so that's why fetal sections are a bit different when you look at them, when you are studying them. Okay, so this is the median section or the sagittal section, which divides the fetal body into two halves, right and left halves. Now this is the, when you are looking from the sides, this is the lateral views, this is the transverse section. Okay, coronal section or the frontal section, I told you already, this divides the body into anterior and the posterior halves. Okay, so this type of section is called as coronal section. Already we discussed that. So that's all for the introduction. Now after till here, any questions? Any questions, any doubts?
Tomorrow we are going to have this in the exam.
Yes. Tomorrow, MCQ test beginning of the class.
Excuse me, ma'am.
Yes.
Ma'am, what is meromelia?
Meromelia is underdeveloped limbs. Okay. Amelia is no limbs. Meromelia means underdeveloped. So that means we have bones, but they are not well developed, very small bones, or maybe truncated limbs. You will get such kind of limbs. These are called as meromelia. This situation is called as meromelia. Okay.
Okay, ma'am. Thank you.
Any questions?
Excuse me, ma'am.
Yeah.
Ma'am, in flat bones, there is no medullary cavity.
Hmm.
So, how does bone marrow formation take place?
Yes. Medullary cavity is not there, but it has got cancellous tissue in between. Okay. So outside, only thin layer of compact bone is present, but inside it is filled flat bones. All flat bones are filled with cancellous bone. Okay. Remember cancellous bone? Cancellous bone means it has got trabeculae, it has got lot of space, a spongy bone, I told you. So in that spaces, okay, the spongy tissue, it is filled with bone marrow, red bone marrow.
Okay, thank you, ma'am.
Yeah. Any questions, guys? Okay, till here clear, all of you? Shall we go ahead?
Yes.
Yes, ma'am.
Okay. Now let us understand some basic concepts related to cell division. Okay. You must have studied this cell division before also, but little bit we will discuss in relation with how our this what to say human development is dependent on. So you'll have little bit more idea or understanding about cell division compared to what you have studied earlier. Now next week assignment is you have to draw mitosis and meiosis and label all the stages of mitosis and meiosis. Okay, I repeat, next week, next Tuesday till next Tuesday. Okay, not Monday, next Tuesday, not this Tuesday, not tomorrow, next week Tuesday. Okay, you will have to submit again in Google Classroom. I'll provide a link to submit your assignment on cell division. You have to draw on a chart cell division, mitosis and meiosis, the stages of mitosis and meiosis, and label them. Okay, properly label all the stages and submit. Okay, submit the copy picture of your chart. Okay, clear all of you?
Yes, ma'am.
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 Now, what is a cell division? Cell division is a process by which a parent cell divides into two or more daughter cells. Now, cell division is usually a smaller segment of a larger cell. Okay. We are going to see only the basics of cell division. We are not going to study mitosis and meiosis today. After this 10 minutes, we will end the class. So we will get back tomorrow. What is important is you need to understand certain structures, certain things before understanding the cell division process. Now, what are chromosomes? All of you know what are these, but we'll just quickly revise what are the structures related for cell division or required for cell division. Okay. Now, this is a chromosome. Inside, where are these chromosomes? When you go into the cell, enter into the nucleus, and there is lot of condensed genetic material, okay, inside the nucleus, inside the nucleolus. Now this genetic material, when it is entering into cell division process, they condense and gives rise to this butterfly shaped or X shaped structures called as chromosomes. Okay, to be precise, chromatids from their chromosomes. Now from this, out of this, we stretch and pull out. We get something like this. Okay, we take out this condensed structure, remove off like a ribbon and stretch it and look at it. We get this kind of double helical structure called as DNA. Okay, which we all know what is this, right? So, what are the chromosomes? These are the structures that transmit genetic information to the next generation. Now, let us see a little bit about the structure of a chromosome. This is a chromosome. Okay, when we pull out, it comes out like this. Okay, double helical structure. Okay, see here how we are going back now. It's a double helical structure of DNA. When it is condensed very much, okay, like a long thread, this thread, if you just go back and see, this thread is winding around some kind of basic proteins called as histones. Okay, histone proteins. So these threads are winding around the histone proteins. So it's not just simple DNA which is present here. lot of proteins are present which are binding this DNA to it at various levels. So these are called as histone proteins. Now, even this included the thread along with histone proteins all combined together. It forms this thick material, okay, which is now winding so much, and this winded structure forms like this hazy structure called as chromosome. And what does it contain? It contains arms. What are the arms? Two arms are short and two arms are long. The short arms are called as P arms and the long arms are called as Q arm. Okay, P arm and Q arm. At a point, all the arms are joined together. Okay, this is a very important point. This is called as centromere. Okay, this is called as centromere. So this is a basic structure of chromosome. Now during cell division, we have spindle fibers joining here, okay, and splitting this chromosome into two parts, okay, this separately and this separately, okay, P and Q on one side and another P and Q on the other side. So this is where the centromere is a point where the spindle fibers are attached during cell division. Now, what is chromatid? Two copies of the same chromosome attached together are called as chromatids. Okay, so these two. Now, see here, let's say this is P arm, this is Q arm. So, when they are attached together, when these are the two copies of same type, two P arms are of same genetic material, okay, two copies of same chromosomes. So, these are called as sister chromatids. Okay, chromatid, the single arm is called as chromatid. So these two are called as sister chromatids. Where they are attached? At the centromere. You can see a specific sticky substance which is called as centromere. Okay, centromere is a primary constriction where the sister chromatids are attached. Okay, this is the point where the kinetochores are present. Again, it's a protein which is present at the centromere, attached to the centromere, where the spindle fibers of, spindle fibers. What are spindle fibers? During cell division, the spindle fibers are coming and sticking to this kinetochores. Okay, so these spindle fibers which are only attached to this centromere kinetochores are called as kinetochore microtubules. Okay, kinetochore microtubules. Clear till here, all of you?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Now, what is a kinetochore? It's a protein structure that assembles on the centromere and attach sister chromatids to the mitotic spindle. Okay, so this is the mitotic spindle that move the chromosomes during mitosis and meiosis, right? Now, what is a karyotype? So, you know about chromosomes, you know about what are the parts of the chromosomes. Now, let us understand briefly about what is a karyotype. Karyotype is a complete set of chromosomes in a species. It describes the number of chromosomes. It's actually a technique where we can study these chromosomes. If we have to get this type of structure, we have to take the cell till cell division, okay, because otherwise normally when cell is in the resting stage we will not see chromosomes. We will see only lot of wavy thread which is called as chromatin, and only during cell division this chromatin assembles together to form chromosomes. Okay, so what do? We use a certain kind of technique, okay, where the cells starts dividing. We will initiate the division process and we will stop them before splitting. Okay, we add colchicine. Colchicine is a dye which stops the cells in the metaphase stage. In metaphase stage, what happens? You will see only chromosomes like this. Okay, where maternal and paternal chromosomes are all accumulated like this. So what do? We just break the cell membrane and plate all these chromosomes on a slide. And this slide, when we keep in a microscope, we get pictures of them, and from their pictures we take and arrange them in an order as per their size and shapes. Okay, so every chromosome has got its pair. Okay, so we have pairs of chromosomes. That's why we say there are 23 pairs of chromosomes. We don't say 46 chromosomes together usually. Okay, yes, whenever we say how many chromosomes, we say 46, or we have to remember there are 23 pairs of chromosomes. Why pairs? Because each pair contain one from mother and one from father. One is maternal chromosome, other one is paternal chromosome. Okay, so human organism contains cells which contains 46 chromosomes. Those are 23 pairs of chromosomes. One set is coming from the mother and other set is coming from the father. Okay, maternal and paternal chromosomes, they're all arranged like this. So see here, 22 pairs are called as, okay, autosomes. Okay, these are normal chromosomes, whereas last one are allosome or a sex chromosome. Okay, chromosomes pair, that is either X and X. If we have two X and X chromosomes, that is the child is a female child. If we have X and Y chromosome in the last pair, so then you have a male child. Okay, so we look at this pair and we can decide whether the child is male or female, whether the fetus is male or female even before birth. Okay, so this process is called as karyotyping process. Clear? Now what is a cell cycle? So we'll not go into cell cycle today. Till here I would like to end the class. So go back and give one reading. I'll send this PDF by evening, and just have a look at whatever we had discussed, and study a bit more about it, and you can complete your assignment of cell division chart. Okay, any questions?
Ma'am,
Yes.
ma'am, why there is a space between the third and the fourth chromosome
No,
and
no, no,
15 and 16 chromosome?
no spaces, dear. It's just how we organize it. We can keep it all in one order also, no problem. Okay, this
Yeah,
organizing
ma'am.
also, we are not